Hello everyone, and I welcome you all once again, all my fellow biotechnicians and all our dear YouTube channel subscribers. Today's topic is common mistakes students do in CSIR NET exam for Unit 1. Well, you cannot deny the fact that this is a very important unit. Why? Because it has relevance with many other units. You have to understand the concepts to understand other unit topics much better. Concepts which are included with Unit 2, Unit 3, Unit 4, where you are studying about molecules, genes. If you don't understand how the proteins work, if you don't understand how carbohydrates, lipid structure is, then it's difficult to follow, if, for example, the membranes under Unit 2 or the cell signaling proteins in Unit 4. It has also relevance with Unit 6, 7, Unit 13. So basically most of the units in CSIR. So the verdict is that you simply cannot skip this unit. No. So let's see what general mistakes students do while either preparing for the exam for Unit 1 or while giving the exam or attending the exam. The first point is not focusing on chemistry basics. Yes, you have to know what is the atom structure, what is ion structure, cation, anion, how are they formed, giving or taking of electrons, what is the molecular structure, what does a molecule consist of. You have to know this concept. Next is elements, their electronegativity property. Why? Because depending on the electronegativity, the chemical bond formation concept is dependent. Not forgetting, you have to also know certain elements in the periodic table. What are their valencies, what kind of cations and ions they are forming. You see, these all form the basis or foundation of further studies. That's why you should definitely pay attention to this. Aromaticity. What is an aromatic compound? What are sigma? What are pi bonds? How are they formed? How does resonance occur? Because if you want to study the absorbance property of proteins, but you don't know what is resonance, you don't have an idea what are aromatic compounds, then how is it possible to go forward? Next is the chemical bonds. Yes, different type of chemical bonds, covalent, ionic, specifically hydrogen, hydrophobic van der Waals interactions. Because if you don't know the basis of bonding, then how are you going to know that, okay, in a water molecule, since oxygen is more electronegative, that's why it is going to attract the electron towards itself and get a delta negative charge. The other end is going to get a delta positive charge. This is what gives the molecule a polarity. So concepts like polarity, hydrophobic, hydrophilic will come from the understanding of bonds only. Next are functional groups. Studying amino acids, studying the classification, different R groups will become much easier if you know what all different types of functional groups are there and what are their structures. Ethyl, methyl, ester, ether, sulfhydryl, phenyl. These all will definitely make your preparation journey much easier, and students jump to higher topics directly without knowing these basic foundation terms. Next is stereochemistry. You need to know the different configurations, L and D configuration, geometric, cis and trans isomer. That is how you will know the peptide plane, right? Oxidation reduction. Yes, you need to know Leo, the lion says Gur. That means oxidation is loss of electron, reduction is gain of electrons. So some logic you need to set. Otherwise, questions like oxidized form, reduced form will often be a part of CSIR questions. pH concept. If your hydrogen ions are increasing, then is your pH increasing or decreasing? A basic question, but I have noticed many students get it wrong. So you have to know the pH concept along with the acid base. And not forgetting, the most important is basic numerical concepts. That is normality, molarity, mole concept, Dalton, gram molecular weight. So these should be at your fingertips and you should be able to do the calculations very easily. Next is studying topics superficially or memorizing blindly. You can't put an entire book into your brain just like that. And that too, students will try to do it, learn, unlearn and revising, not revising, trying to memorize it by heart, just by reading it out aloud, reading it multiple times. But that's not going to help until and unless you understand a topic. I don't want you to miss out on the detail. In fact, that's the mistake what students do while preparation. I will take as an example amino acids. So what all things students will generally prepare if they are preparing by themselves? Okay, they will know the classification, what different types of amino acids are there. They might know something about the PI concept, pK values, right? And something about the function that it forms the protein. But there are many aspects which you should learn under amino acids. Yes, classification, that's fine. But did you know there are many types of classification? This is detail based on R group. That is the first classification. Based on the essentiality, that is where you have essential, non-essential amino acids which can be synthesized, cannot be synthesized. Third is what happens to them ultimately, whether they become glucose molecule, they can give rise to glucose molecule, or they produce ketogenic molecules. So based on that glucogenic, ketogenic. I hope you're getting the difference. Amino acids, they would be present as L and D form, that they are mirror images of each other, that they are enantiomers. So which one is preferred mostly in proteins? That is the L form. These all small details make a lot of difference in CSIR preparation. Third thing is titration curves. Yes, so titration curves, you're learning the pK value, you are learning the PI value, that's okay. But are you able to connect it with the pH concept, with the Henderson-Hasselbalch equation to calculate the pK values or the concentration of proton donor and the proton acceptor? Are you able to do all this? So this is where you have to concentrate more, and last but not the least, you need to know how to calculate charge on an individual amino acid. Not only you need to know the pK values of the R group, but also what will be the charge of an individual amino acid, or in fact on even a peptide molecule, or what will be the PI value of a peptide, of an unknown given peptide sequence, which is simply depending on what all types of R groups will be present. Third concept is, it's a tough unit, you may need hand holding. Yes, students. So as I told you, this is a very conceptual unit. Just by reading the books, you cannot get the concept. Your chemistry basics should be clear. Your lot of detailing should be there. And to be honest, in 6 months, doing it on your own can be difficult. So what you should rely on? You should rely on genuine online classes who will bring the concepts absolutely crystal clear to you. Make it in short, concise manner, and also provide some crisp details of them which are important from the exam point of view. Plus, there should be some kind of doubt solving in case you are getting, while studying you will get lot of doubts. So for that also, there should be some instant doubt solving, that okay, I had a doubt, I took a picture, I mailed it, and you should instantly get your answer, so that your studies are not stopped at any point. Then you will get all the recorded lectures you should have somewhere, because constant revision is required, right? So better if you can get the recorded classes of whatever lectures have been done. And also you should be revising, have a provision for revising constantly and getting monitored through exams, right? And last but not the least, you need some study tools, like how the study and recollecting, retaining concept can become better, along with some tips and tricks like some mnemonics. Right now I just told you one for oxidation reduction, right? So these all you will get, you need to have a little bit of hand holding. That is when students try to prepare it on their own and may face some issues. So you know where is a genuine authentic source. You decide on your own by the excellent track records. And yes, at Biotechnica, we provide all this. So you should go for something where you are getting all of this, because that will constantly monitor your progress and help you prepare very efficiently. Next common mistake is not preparing in a systematic way. Well, if you take a pathway, let's say for glycogenesis or glycogenolysis, that is glycogen synthesis and glycogen breakdown, right? So you have to learn many details in a systematic way. First thing is, what is this pathway for? Okay, so glycogenesis, if I have glycogenesis and I have glycogenolysis, right? So another thing is that you need to know, you need to break the words and study. Many of the terms and terminologies, they are based upon the word. Glycogen is the molecule, genesis means synthesis, and lysis means breakdown. So glycogen breakdown and glycogen synthesis. You need to know what is that pathway. Then you need to know why this pathway takes place. Okay, when too much of glucose is accumulated in the cell, then it goes for synthesis. What will you do if you have too much food in the house? You will eat some, you will store some. When will glycogenolysis take place? When the food source is less and you have to break down the stored food to get the glucose. So why this pathway takes place? Third thing, so this is the systematic way. What are the details or what are the steps involved? Like the ultimate steps, like what it binds to glycogenesis, for example, it needs to bind with a UTP, right? Associate with a UTP. All the steps which are involved in a pathway, all that you need to know. What is the starting pathway? What is the product of the pathway? These are the details, okay? Including the enzyme names. Not forgetting, very, very important, the enzyme name along with what cofactor it is requiring. Next is when this pathway is happening, okay? When, why, more or less same. Where is this pathway happening? What is the subcellular location? Or if at all, which type of cells? Okay, so here there is detailing also which you are understanding. Very important part of unit 1 which students miss out is regulation. So again I am telling you it's about the details. You may study the pathway but you are missing out on the regulation, which itself is a target for CSIR questions. Okay. Sixth thing is, how is this, so basically what are the activators, inhibitors involved in regulation? Those things you have to study. In fact, even hormonal control. For example, glucagon, epinephrine, do they activate glycogenesis? Do they inhibit glycogenesis or whatever? Or else if you have insulin, which is released when there is high concentration of glucose. So what they will do? These all things you need to keep in mind. And last thing is about the bioenergetics part, right? So how much ATP is produced, whether they are exergonic pathway, endergonic pathway, and whether they are happening spontaneously or not, how much ATP is released by which molecule, which is the ATP contributor. So I hope you are getting the aspect of how systematic way preparation. This way you are not going to forget any of the concepts of this pathway. It's going to be crystal clear. Maximum, if you have to study these two pathways, it will maximum take 1 hour with guided studies, right? Next concept is not making proper study tools. See, you will not get totally consolidated tools anywhere. Biotechnica does provide revision tools for easy access. So difference charts and concept wheel, all concepts together in a place. But I will suggest you also make your own separate revision tools, study tools, right? For example, let's say what is the enzyme name or what is the protein name and what type of cofactors it is having or what type of prosthetic group it is having. Okay, so let's say you have hexokinase, which is the first enzyme in the glycolytic pathway. So we know that the cofactor required for this is Mg2+. Like this, if you can keep on entering the details whenever you can for whatever step you are studying, it will be really beneficial. Second thing, what you need to have a note on yourself is the different formulas. Okay, for different terms and terminologies, what are the formulas? Make a note of them and keep it separately. These are tools which will be necessary later. All the pathway details which I already mentioned, along with which enzymes are inhibited, activated, and all possible details. So this you can make in the form of a table, right? So this is what is systematic study. If this is missing, then you can terribly fail in answering the questions in CSIR. Next thing is pK values. For amino acids, the side chain pK values, yes, you need to write down somewhere. Okay, glutamate, it has a pK value pKr of 4.3. Aspartate, it has a pK value of 3.7. Whereas lysine, it has a pKr value of 10.5. Yes, these are going to really help you and you need to write this down. Not forgetting in the same concept, the single letter codes for amino acids. Many students will make a mistake in that, but in several unit questions, you will find a protein sequence given in single letter code. And based on that, you have to predict on the properties. So ensure that you also make a note of them. Then very important, enzyme inhibitions. So these are like must revision tools, study tools. You must have it with you. Like what happens to the Km for a different type of competitive, uncompetitive, non-competitive. What happens to value of Km? What happens to value of Vmax? Draw the Michaelis-Menten graph. Draw the Lineweaver-Burke plot. Okay. So all these things must be at one place so that you don't have to search it separately for all the time. And lastly, differences table, which already Biotecnika provides in the form of concept table. So differences, for example, like that in ABZ DNA, right? So those concepts, every time they will ask the sugar puckering, whether it is endo, 2-endo or 3-endo, or they will ask about the pyrimidine and purine bases, whether it is anti, whether it's syn, and further many differences in detail, lot of concepts are there. Right? And also you need to know about the protein conformations. That is, what is the dimension of alpha helix, what type of amino acids can be there, or beta sheet, what is the dimension, what type of amino acid. These are like making tools, keeping it in one place for not only understanding but also revision for later use. Sixth point is not focusing on units in numerical problems. Yes, you should have your focus on topics like, let's say the question is asking you out of a DNA, like if you have to code for a genome in the DNA, so a protein is made out of this DNA. And if you are given a protein and you are asked that what will be the length of the gene coding for that protein. Now you have calculated, we know the length of the gene is 3.4 nanometer. And we want to get our answer, we have got our answer in nanometer. But what if the question in a very small line, it has mentioned in micrometer, which you haven't noticed. So that is where you can get a mistake. This is while performing in the exam. Otherwise, you need to know the differences. Let's say the enzyme units, okay. So whether you are getting the enzyme unit or else the question has asked in enzyme unit, but you have answered in Katal. That is again where you need to know the units of entropy, that is enthalpy, entropy and Gibbs free energy, specifically gas constant value, etc., Faraday's constant, exact units you need to know, right? And also the enzyme activity, I'll tell you a small difference, like when you take in enzyme activity, it will be Katal, if you want to give it in Katal, Katal is mole per second per kg, okay? But when you take in enzyme unit, the specific activity, it will be micromole per minute per mg. So as you can see, there is a fixed thing. Mole has to go with kg, second has to go with kg. You cannot do opposite. But when we don't write and practice, when we study haphazardly, if someone doesn't point it out to you, these are the common mistakes which you can do. Last 2 points, doing selective study. Yes, you cannot pick things from the syllabus. Okay, I am very comfortable with enzymes. I will study enzymes, but I will not do the kinetics part. I will study only the classification. Then what is going to happen? No, if you don't study the kinetics, mostly questions will be from kinetics. Okay, what if that time question is not there from inhibition at all, and all are from kinetics? You thought that okay, I will do proteins and amino acids, but then I will not concentrate on carbohydrate lipids. To some extent you can be saved because these are lesser important topics, I will say. Proteins, amino acids are more important topics, but you do get questions from carbohydrate like epimers, anomers, from lipids also, membrane lipids, phospholipid structures, archaebacteria, normal bacteria, you will get all these differences. So never do selective study. If you are preparing a unit, then prepare it completely. Then only you will be able to answer at least 90% of the questions which come in the exam paper. Last topic is unnecessary focus on not so important topics. For example, you have amino acid metabolism, right? Or else you have vitamin metabolism. So all these metabolisms, they take a lot of time to study, and honestly they are not so important, okay? So you have lipid synthesis, lipid anabolism. These topics are not so important, but students spend lot of time sometimes to study all the different amino acid metabolism. So clearly you can avoid this and you can definitely skip all this. So I hope this concepts were beneficial to you. What are the mistakes? And I sincerely hope that you students will avoid these mistakes while preparing for CSIR exam. Okay. So all the best to everyone. Do subscribe our channel if you haven't already, and mention in the comment section if you like this video, or else if you want any particular video specifically. Thank you everyone for your time and attention.